Welcome back, everybody. It is 11:30, and before we get started with the Q&A and wrap up, I just want to say thank you to our two college mentors who were in the breakout room assisting, Bryce and Annie. Thank you all for joining, and I heard you guys had a fantastic time, and there were some great questions from our groups. And it is time for our Q&A and wrap up with our two college mentors who are going to introduce themselves, and Betsy's going to assist with leading this, and then it's going to be time for you guys, students, teachers, get to those keyboards, get to those cameras, turn them on. We want to hear from you. Teachers, you can type questions in chat for our mentors and our presenters today. So please do not be shy, but I'm going to stop speaking and let our amazing presenters for our Q&A and wrap up, Sinai, Dylan, and Betsy, take it away.
Yes. Oh, I can't wait to get to the part about the students. They had some amazing ideas in the chat and in the breakout room. Dylan, you want to go ahead and introduce yourself?
Yeah, of course. I'm excited to see what you guys have done. So hi everybody, my name's Dylan. I'm a senior from the University of Notre Dame. I'm a computer science major. So a little different from this, but we will be able to talk a little bit about how you can kind of combine a lot of what you guys are doing with different fields of engineering, which is pretty cool. So my interests in engineering right now are data engineering, artificial intelligence, AI. So if you've used ChatGPT or any other form of AI, everyone uses it now. That's kind of a lot of the work I do. I've been with Engineering Tomorrow since my sophomore year of college, and I'm super excited to be with you guys here today. I'm going to jump into a little bit about my experience and what I've been up to. I got a little bit about what I did in high school. So I'll start off there. When I was in high school, I graduated in 2021, so right after COVID, which was kind of crazy. But in high school, some engineering related things. I started off as a robotics team captain. I joined that team my freshman year, and worked my way up to be our team captain. I went in knowing absolutely nothing about robotics. But that's really what made me fall in love with engineering. In my school also, I founded our coding club, and I also participated in a biomedical engineering event in New York City called Perry Outreach, which is a really cool way for high schoolers to learn a little bit more about the medical field, but more specifically the biomedical engineering field, where we worked with a bunch of different types of biomedical engineering projects. We got to we kind of learned how different medical devices are made. I also got to put a cast on my friend, and she put a cast on me as if we had fake broken bones. It was a lot of fun, and it was a really cool way to learn about biomedical engineering. Then once I got to college, I ended up kind of shifting gears towards computer science. So I did my freshman year, I worked on a project where I worked with a Disney Imagineer on an animatronics workshop where we taught middle schoolers and elementary schoolers how to build a very simple animatronics setup. And that was very cool. Like I said, I'm very interested in AI. So I did AI research my sophomore year. I worked on campus with a professor over a summer. It was a great way to get some experience when it's kind of hard in early years to get an internship. So I was able to do some research. It was pretty cool. I got to learn about kind of how AI works, and it really helped me for later classes that I'm in now, where I'm really learning the inner workings of machine learning and AI. And then my junior year, second semester, so last semester, I did a study abroad program in Hong Kong. I was an exchange student for a semester all the way over in Asia. It was very cool experience. So if you guys have any questions about study abroad, feel free to ask us that. I can answer any questions you guys might have in the Q&A part. And now most recently, this past summer, I had an internship in data engineering, where I kind of worked between using a lot of code and programming and software, but also integrating data into that to make it more efficient for my company to use. And now once I graduate in 2025, I will end up going back to that internship full time.
That is so exciting, Dylan.
Yeah. Oh, I didn't realize I think I left the animations on here. Betsy, if you could click a couple times just to show maybe one more. There you go. That's it. Sorry, I had animations on here. I forgot that I had those. But here's just like a couple pictures of things I've done throughout my experience. So in the top left, I've got a picture of me at Perry Outreach with that cast on my arm that my friend put on me. Cool way. I got to wear scrubs and everything. It was very fun. And then there's also a picture of me with our robotics team. We got a trophy one of the years that we were competing. So that was really cool. We built a robot and competed against other schools in the area. Then I moved on to that top right side where I was working with that Disney Imagineering workshop. So there's a picture of me working with some of the kids we worked with. It was very cool kind of first college project experience at Notre Dame. There's also a little picture of the dome at Notre Dame, which you can see behind me in my image or my video. And then also, I just had a little screenshot of code. I have a lot of screenshots where I'm just taking a picture of my computer to remember what kind of code I'm working on and kind of scroll back and forth. But just a little taste of one of many lines of code programs that I've written. Then you gotta go on over to that middle picture where I was doing my study abroad. So that's a picture of the campus I was on. It was right on the water. It was very cool. I got to do a lot of travel, but also I met a lot of really cool people who were also engineers over in Hong Kong. I made a lot of friends from not just American exchange students, but European exchange students, local Hong Kong students, other students from Asia who were exchange. It was very cool. And then lastly, on the bottom left, I have some pictures of me at my internship. I was in Chicago. I made a lot of really cool friends there, and internship is a really great way to meet other people who are going into the field, who are also, your age, trying to figure out what they want to do with their career. So there's just some fun pictures of me from my data engineering internship.
Excellent.
Yeah, that's great. But hello everyone, I'm Sinai. I'm a mechanical engineering master's student. So I did graduate in 2024 with my undergrad environmental engineering, but currently I'm doing mechanical engineering. So some of my interests include biomedical, biotechnology, engineering, as well as mechanical design and sustainable development. And yeah, I'm currently at Cornell University. And a little bit about my background. So yeah, I'm going to do the same thing that Dylan did. I'm going to start a little bit with what I did in high school. So I was involved in a STEM UC Berkeley program in California. I'm from California, but yeah, it was called Summer Math and Science Honors Academy. And there I was able to build a lot of my engineering skills, learn about engineering, learn about the different types of engineering that I never knew about. And getting into college at Cornell, I realized I really liked environmental engineering. I wanted to work with wastewater treatment, and I joined a project team called Agua Clara, and they essentially focus on providing water energy resources for communities in Honduras. So that was a great project that I got involved with. And during my involvement there, I did a lot of manufacturing stuff. So I built a lot of things, and that kind of sparked my interest in mechanical engineering. And throughout my summers at Cornell, during my undergrad, I did a lot of internships with biomed and pharmaceutical companies. So there I really got to experience what it is to be there, the type of work. I did a lot of manufacturing work as well as environmental health and safety stuff. So I got the interest from mainly internships and some classes that I took. So I decided to pursue my master's early in mechanical engineering, and currently I want to go into the manufacturing world in biomed and biotech. Next slide, please. So yeah, this is also, I guess, like a little roadmap of timeline, pictures to show that. So on the left corners are just like projects or things that I've like presented about and worked. All these projects I've always worked with people. I feel like engineering is always collaborative, which is really nice because then you can combine different ideas. I also interned at DoorDash and Zymergen early into my collegiate journey. There I just learned how to like really collaborate, hone in those engineering skills that I had. At the top, kind of right, you can kind of see that's at the wastewater treatment plant at locally here in Ithaca where Cornell is at. And there, as you can see, I was very much in there trying to build stuff. So it was very great to experience that. And then later on, I did a lot of biomedical world on the top, or biomedical work on the bottom corner right. Is a device I helped build, used in a hospital, and I was able to see that. So similar to Dylan, I was also in scrubs in the workplace. So it was really cool. And for my master's project, I've worked with car engines, which you can see in the middle bottom, and as well as like done research with biomechanics And on the left bottom corner is a device that I helped manufacture. On the right of that, you can kind of see me with the lab coat. So that was at my internship last summer where I did a lot of compliance stuff for a pharmaceutical company.
Man, you guys have some great backgrounds. It is so cool. I can't wait to hear more when the students are asking you guys questions. So now you've learned a little bit about Dylan. You've learned a little bit about Sinahi. You know a little bit about me from before. We're here for you guys, and we cannot wait to hear how your particular labs went. I want to give a quick shout out. I know we talked about things being collaborative, but Renaissance Academy came up with a great idea. In all of my years of doing this particular lab, I had never had students offer to link their blasts so that other students could use those results. And guess what? We tried it out today and it worked. So those of you that had that idea to help your other colleagues, what fantastic idea. Renaissance Academy, high five to you and your students for that. And to take us over, I think Jerry Dawes is out there at Academy of Mount St. Ursula. So I can't wait to see him and his students. How are you guys doing out there?
Can you guys unmute at Academy of Mount St. Ursula? Oh, there we go.
Yeah. Hello.
Hi.
Betsy, how are you?
I'm great. How are you guys?
We're doing great. And we have a veteran here, Dr. Suraj Tiwari, who's been with ET since the inception.
Hi.
Awesome. Good to see you. Thanks for being here. So Jerry,
So
do you
whenever-
guys have students that would like to
Whenever-
talk with us?
Whenever you're ready, we're ready.
I think
ready
we're
for
ready.
you.
Let's go.
Come on
Yeah,
up,
let's
students.
see it.
So
And
if
as
you
those
guys
students
come on up,
come
come on
up.
up. Come up in the camera. If you guys could just introduce yourselves, give your names, and just talk about your experience. Can you see us, Betsy?
We sure can. You
Yeah,
guys
we
look
do.
great.
Okay. All right. Go ahead, guys. Give your names.
I'm Zenobia.
You got to speak up. Please speak up.
Okay. Get closer, get closer. I'm Zenobia.
I'm Kayla Robinson. I'm Kayla Montano.
And
I'm Malika.
Oh, it's so nice to meet you all. How did the lab go for you guys?
it was fun.
It was a cool experience,
Mm-hmm.
for sure. Yeah.
Do you,
What was
were
your
you
favorite
able to
part?
get the diagnosis?
Yeah, we did patient X. Is there like a specific sequence you want to see or like you want to hear about, or is like everything?
You could tell us what were your most like, what did you like most about this lab?
We liked putting the thing in the blast, and then we thought the structures like of the different protein sequences, those were cool. Like the screenshots of those were cool, and then finding out the different diseases like attached to each protein sequence.
Awesome. Oh, wonderful. What are you planning on doing when you go to college? Have you thought about it?
I want to go into psychology.
Anyone else
Anybody?
have any ideas?
I want to go into neuroscience.
Awesome.
I want to major in English.
Awesome. That's good.
Oh.
All right.
Oh.
I love that you guys are thinking about what you're going to do next. It's so good. So good. All right. And it sounds like you were able to run the protein sequences and get your blast results. Is that right?
Yes.
Awesome.
Very good. Well, thank you so much. I am so proud of you guys for being the first to share. High five to you.
Yeah, thanks for coming up, guys.
Thank
All
you
right.
so much. Do you have any questions for us at all? Maybe for Dylan or Sinahi?
No, thank you.
No? Okay, wonderful. Thanks so much.
Betsy, I'll be hanging out with the class right after, so I will be answering all of the follow-up questions. But we do have some other students that want to present as well.
Oh, please
Awesome.
come on up.
Great.
Tiwari, please bring up who are the other students. All right, so they're coming right up.
Sounds great. Thanks so much, Jerry.
You're welcome. All right, if you guys could just introduce yourselves, give your
Hello.
names, and just talk about your experience with the lab so far.
Hi, I'm Alexa.
I'm Jamila.
The lab, we did have some issues running the blast. We definitely had to run it twice because the results are really slow. But for the most part, we did end up getting our results and then finding out the diseases of like each patient, and that was pretty cool.
Yeah, we found out that a lot of them were related to cancer. So a lot of the diseases that came down were mainly cancer related, or like anemia and insulin. Yeah.
Very good. Yeah.
And I got to give a
That's
plug
cool.
here, Betsy. Jamila was probably one of the first students who got this right. Right after you finished presenting, she was holding her hand up and letting me know she got it.
Oh, that's
That's
amazing.
amazing. Good job. That's so wonderful. And I love that you were following along so well that you could get it so quickly. I'm curious, when you guys ran the blast the second time, did the second result come back before the first one, when it was taking a really long time?
We actually did it on two different pages because the first one it was through UniProf, but it wasn't coming out right away. And then we did it on the medicine page, the one that's on slide 23. That one.
Yeah.
Okay, perfect. Oh my goodness, that's wonderful because we can give those tips to other students as well. So I'm so thankful that you guys shared that. Thank you so much.
Yeah.
Anybody else something to learn? All right, so we're going to allow another school to present at this point, Betsy, but great experience here. Thank you.
It sounds wonderful. Jerry, thanks so much for being there and representing.
Yeah, thanks guys for sharing.
Thank you, Jerry. So we have another high school. They just turned on their camera, then they turned it off. I spotlighted you guys. Rouse High School, Texas. I have you guys spotlighted. You guys need to turn on your cameras. It was just on. There you guys are. Hi.
Hi.
I see you guys ready to present? I don't think we can hear you. Oh, there you go.
There we go. So tell us, how did it go? What was your favorite part?
Really well. I enjoyed like being able to I just thought it was really cool how like the program could like just take like the sequence and then come up with like results. I thought that was really cool.
Oh, isn't it amazing? Did you think today when you woke up that you would be out there looking at amino acid sequences and diagnosing patients?
I did not. I didn't know what to expect walking into this lab. But I'm really
It's how incredible. That's awesome. That's so wonderful. So I'm curious, did you like diagnosing the patients? Did you like using the tools? Did you like learning about DNA? Of those three, what were your favorite parts?
I like using the tools. I enjoyed yeah, like I said earlier, I thought it was really cool how they get worked. I thought it was yeah, it was fun to like try yeah, and I also liked diagnosing using the tools. I thought that was pretty cool.
Fantastic. Wonderful. Well, what are you going to do when you graduate? Do you know?
I have a rough idea. I mean, I want to go to college and major in like biology or something because I do want to go into the medical field. So don't know where yet. Don't know where yet, but we'll figure that out soon.
Oh my goodness, you're going to do amazing things. I can tell already. Thank you so much for sharing with us. Do you have other classmates that want to speak as well?
Thank you.
No worries. Don't be shy. It's all good. Thank you so much.
All right. We have Lancaster High School who had their hand raised. I'm going to spotlight them if they can unmute. Oh.
I also see some hands raised at Catalina. I'm not sure if they want to go next. Is that right? Okay. So after Lancaster.
We've got so many people coming up. This is awesome. Hey guys. Do you want to introduce yourselves, say your name, and then talk a little bit about your experience? Oh. Can't hear you guys.
Can't hear you. I know they're on, they're not on mute, so I'm not sure why we can't hear them. Oh no.
This is like real-time engineering, troubleshooting, audio. I get it totally. That's what we do as engineers, right? We troubleshoot.
Good job. While we figure out technical difficulties in the chat, we had
I see Science Park High School seems ready. All right, let me spotlight Science Park High School. Can you guys unmute?
Oh, this is the one we're gonna want mute.
We might have gotten Lancaster.
Lancaster. Okay, we'll go back to Lancaster and then Science Park. Let's see. There you are. Now we can hear you.
now.
Okay, hi, I'm Kaylee. I'm a senior. We also had trouble with Blast, but Jenna's hers worked and everything, and then we were able to like find out the diseases. Yeah. I'm Jenna. I'm Kaylin, go ahead.
I'm Morgan. Yeah, I think it was good. The Blast was good. Some of us had already had experience using it in different classes. So that was good to like already kind of know how to use it. So that helped.
What kind of classes did you guys use it for already?
So we used it last year in AP Bio to do like the same thing with like human diseases and stuff like that.
Yeah, that's super cool. It's a good shout out too for, you know, maybe not every school does that, but just to know, like, if you're looking for classes to take, if you're, you know, underclassmen and trying to figure it out, AP Bio is a great option for that.
Yeah. Totally. And you guys, oh man, your teachers at that school are rocking it, because I never even heard about it until I was out of high school. And so the fact that you guys are doing this now, you are all way ahead of the game in these areas. So congratulations to you and your teachers too.
Thank you.
Thank you.
Yeah, thank you. Such good energy. I love it.
We have Science Park High School who was ready to share. Science Park, are you ready? Please unmute so we can hear you. Oh, can't hear you. Audio is going to be our fun time today in Zoom, apparently.
Hello? Can you hear us?
Yes.
Come over here. Come over here. Come over here.
Hi.
We thought we were being fancy, but we're not fancy.
All good, all good. Glad we can hear you.
All right. So one thing I found interesting in the lab, was that we were able to look at, because for my group we did the X one, we did sequence 3, and when we did it, I found out that they had breast cancer, which was also related to, I don't know how to pronounce it, but I think it was it started with an N. I'm sorry, I don't remember it. But I just thought it was interesting using the sequences and using using the program to actually figure out how it worked and everything. I just thought it was neat.
Did you have any difficulties coming up with a diagnosis, or were you kind of able to get there once you got that blast working? Oh, it looks like they muted again.
Yeah, and it was so cool that they were able to figure out pretty quickly. It sounded like that, which kind of cancers were related to those sequences as well. Way to go, you guys.
Yeah, great job.
Well, we have Catalina, Foothills High School, who wants to share next. And then I think Monsignor Farrell had their hand raised after. So Catalina, can you guys unmute so we can hear you guys?
Hello.
Yep, you're good. We can hear you.
All right, so what I thought of the whole experience, I thought it was pretty cool to see all the diseases in the it was really I don't know, it was just cool to see all the functions of the different proteins, and it was difficult. Sorry,
No pressure. I know it's a little strange talking to a whole bunch of people. Don't worry.
I'm good now. The only difficulty I had was looking for all the different diseases, because I didn't realize how many like different proteins can produce the same or different diseases, like cancer and all these other diseases. And yeah, I just thought it was cool.
Isn't it fascinating, all those different like mutations that can happen? And it's amazing when things are working normally, how lucky we all are, right? When you consider all the different mutations. So thank you so much for sharing. Do you know what you want to go into when you graduate?
I'll probably go to trade school and be a plumber or something.
Okay. We need those things working, right? Well, thank you so much for sharing with us.
All right. Thank you, Catalina. Awesome job. And Monsignor Farrell has someone ready there in the front to present. If you guys can unmute so we can hear you guys. There you are.
Nice to meet you. My name is Joseph Catania, and this lab basically just really helped me prepare. I want to go into occupational therapy when I grow up, and I think just getting exposed to this biology and seeing how these proteins go into these viruses, it's very it's very useful, very major.
Nice.
Yeah,
That's
definitely. Super cool to like learn about this even in high school too, and kind of get an idea of how these kinds of processes work through, a one-day lab that you're going to do. But in the future, definitely that's going to be super useful for once you get to college and kind of start taking those classes that are going to be more geared towards that medical field for sure. What was the hardest part for you guys?
Actually working the website. I think that was my
Yeah.
biggest struggle. But once I got it working, I flew through it. I was all good.
There you go. That's amazing. That problem-solving mindset.
Nice job.
I don't have another group that has requested to share yet, but I want to say thanks to all those groups that did come up and share. That was amazing. I want to remind everybody the chat is open. So teachers, if you have any questions in your classroom, please feel free to type them in chat, and our mentors and of course, Betsy will be happy to answer them.
Yes, for sure. So we have a slide of just some questions that you can think about. But we also had a shout out to Dublin High School, Lancaster, Renaissance. You all were asking questions for our college mentors, and that was, what steps should high school students take to prepare themselves for fields such as projects, classes, majors? What should they be doing? So Dylan and Tina, do you want to take those?
Yeah, for sure. I mean, I can kind of jump into kind of the cross section of, I heard there's a couple seniors out there. People are still trying to figure out what they want to do. Some people have an idea of what major you might want to do. I'm not in the medical field necessarily, but I know with engineering, that's definitely, I have a lot of friends who are looking to go to medical school from engineering, who are also doing pre-med along with that. I also have a lot of friends who are pre-med looking to go to medical school. That are kind of all in the bio side, neuroscience, psych side. So there's a lot of different directions you can take with this. But for me, at least, I went into engineering. I knew I wanted engineering. So at least in high school, I talked a little bit about some of the projects I did, finding programs that you can participate in if you want it to be related to medicine or something along those lines. I mentioned that Perry Outreach program that's in New York City. Super cool opportunity to learn a little bit about how biomedical engineering works. It was a one-day thing that I went to. So looking for programs that might be in your area or that your school kind of advertises or sends students to. Maybe talk to upperclassmen or your friends who might have graduated already and see what they did. For me, at least in terms of like in high school, I took as many AP classes as I could. We talked about AP Bio out there a lot. I took AP Bio, AP Physics, AP Chemistry. Kind of if you're looking for engineering or science, those science APs are going to be super important. Also your AP Maths, like I took a BC Calculus my senior year. So kind of looking towards specific classes you can take. Those higher level classes are going to give you the experience that will make your college level courses a lot easier and sometimes even place out of those freshman year, maybe even sophomore year courses, depending on what credits your potential college might take. But otherwise, like looking for clubs that interest you. For me, I had no idea what I wanted to do. I joined robotics my freshman year, fell in love with it. That I didn't know engineering at all. I didn't really know what engineering was. I went into high school thinking, oh, maybe doctor, maybe lawyer, whatever. But I think by joining clubs that really interested me, that really was what pushed me in the direction of engineering. Snehi, I don't know if you have what else do you want to add to that?
No, that was great. I feel like definitely I would second the clubs thing. I definitely think like getting involved with any different interests that you might be exploring. I even if you don't want to go into the biomedical field or the med field, I know this was more like bio and stuff like that, but even if there's like any type of interest that you're or anything that you're really interested in, I would say like explore it. So let's say you're really interested in maybe like environmentalism or maybe space. Like maybe you could find one of those clubs. I'm sure there's a lot of resources in your high schools, but I know there's also outside programs during high school that could kind of expose you to those things. So for me, like I didn't even really get into mechanical engineering until the end of my undergrad. I didn't know that's what I wanted to do, but it was when I joined clubs, when I did those internships, that's what really put me out of my shell. And also just exploring the opportunities that your high school has, in terms of like those clubs. You could definitely get involved. You can talk to people about it. You can get their thoughts about it, and I think that's very important because you might not know what might come up, unless you hear it from someone else. And honestly, you can't know about it unless you experience it. So I would say just put yourself out there. Making sure you get involved. Don't hesitate to explore different things, even if you're like, I don't think that's right for me. Like, even if you're a little bit interested, I would say just explore it for sure.
Oh, well, those are great answers. And going back to finding things you love, you might love space and you might love biology, and you could be the people that are experimenting on those twins to find out the differences that occur when you're in long duration versus short duration. So, find what you love and run with it. I love those recommendations that both of you shared. All right, I got another question for y'all. What was the toughest part about transitioning from high school to college?
Yeah, I mean, I think for me, I know Betsy, I think you have a slide next to this also that's very similar to my answer. But I think figuring out time management skills is the biggest transition because in high school, you go to school early in the morning, you take the bus, you drive yourself, whatever it is. And you get there super early, you're in classes all day, you have a lunch break. It's kind of scheduled out for you in the periods that you have. And then after school, you go to sports, you go to clubs, you go home, you do homework, you go to sleep, you do it all over again. In college, your schedule is a little different. You have a lot more free time during the day, and a lot more freedom with your schedule in terms of picking different kinds of classes and picking things that are interesting to you. In high school, you have a lot, everyone's kind of, for the most part, taking the same things. In college, everybody's taking something different. So I think that transition of going from high school where you have kind of a schedule that's laid out for you, and then in college trying to figure out, okay, I have all this free time. Am I going to go home and take a nap, or am I going to do some homework? When do I have time for lunch? Did I schedule something I did freshman year? I think my first semester, my Monday, Wednesday, Friday classes, I put them back to back to back to back. So I didn't have any chance for having a lunch break. I just went from class to class to class with no lunch break. So kind of keeping in mind those things about time management and figuring out when it's like how much time you need to spend for studying and homework, but also kind of planning, okay, I have all this free time. How am I going to make time to see my friends? And a great way to do that is to form study groups. Or, if you need a break, go see your school's sports teams and go see events for that. I go to Notre Dame. We have football every Saturday. We're doing really good this year. I think we're 6th in the rankings right now. So super exciting. But just finding ways to kind of manage your time between, you don't have your parents there to kind of tell you where to go and what to do. You got to keep track of yourself, when to turn in homework, all of that kind of stuff. So that's kind of, I think, the biggest transition is figuring out time management and figuring about what system works for you.
Yeah, I totally second that. I guess to add on to that, there is a lot of freedom in terms of what you can do. There's no one telling you like you have to go to class, you have to turn in your assignments. If you do not turn in your assignments, you will just, not get that good grade for it. So there's definitely a lot of freedom, but that takes some organizing and some responsibility, I think, for me. In order to organize myself, I had a Google Calendar. I scheduled in lunch, like Dylan said, like you have to make sure, okay, I have to eat, I have to sleep good hours. I have to study for this exam. So I scheduled a lot just so I knew I was kept on track. But yeah, I know Capital City High School has maybe some questions. So if you want to go ahead and unmute, we're definitely here for you.
Also, for anyone else, if we want to do the same format we did for kind of talking about the presentations, if you want to just raise your hand, we can call on people too.
Okay. Hi there, Capital City. Do you have a question that you'd like us to answer?
Yeah. Good morning.
Hi.
My name is Ronnie, and I have a question for you. In your opinion, should we have genetic testing done on ourselves if we have an hereditary disease that the scientists and the doctors say that it cannot be cured? What is your opinion on that?
Wow. That's a really great question, actually. I feel like it might vary person to person. The thing I would say is science is always growing. In our age, like there's so many new medicines, new technology to cure anything. So I would say genetic testing, like if you want to get tested, I would say, yeah, you never know what might come up. There's always, engineers, scientists, like PhD people that are always studying these things. They're always studying genetics. They're always studying, cancers, how to solve them. So you never know, there might be a chance. But yeah, I would definitely say, yeah, great question.
Yeah, I mean, I'll add a little bit to that too. I did a 23andMe, not exactly, the total same thing, but doing that to kind of see what pops up and kind of confirm some things I already knew about myself. But who knows what could have shown up? It could have shown up something that I didn't know was there inside of me all along. So definitely, I think it's as someone who's in engineering and who's super interested in progressing science and progressing research on things. I think, like Sneha said, like it's everything is growing so much in this field, and there's been so much research continuing to grow exponentially. So for you guys also in high school, as you're getting to college, there's going to be a lot of opportunities to join in on some of that research. You'll meet some professors who are doing some really cool, really groundbreaking research that you might get the opportunity to get in there and do some of the work. Even if you don't feel like you know what you're doing totally, you could even get your name on research papers. I was told that in the future, the research I did has the potential to be published, and my name would be published in a paper. So getting involved in that research on genetic testing would be super cool if that's what you're interested in as well.
And I see another question in the chat from Union City. How difficult is it for you to balance your academics with your extracurriculars, sports, or other programs and clubs?
Yeah, I guess for me, I'm involved in Agua Clara, so the project team, but also I don't do sports, but I do dance. It's not really considered a sport, but I'm part of several dance teams. So I definitely, like I said, I use Google Calendar religiously. I'm always looking at that, making sure I'm on top of things. I think the main thing is just putting in that time for yourself. So with engineering, like it could be very time consuming, but just making sure you prioritize, like, maybe I should have some downtime during the day, or maybe I should take a nap because I didn't sleep too well. Like just kind of planning ahead is very, very important. Things can come up and you never know. So I would definitely, I think that was the most difficult part, realizing like, wow, I have no one to remind me I should go to sleep early, or I should wake up early and eat breakfast. Like I can't skip, breakfast. So I think that was the most difficult part, but it does get some getting used to, but I would just say like staying organized, keep staying on top of things. Sometimes you can't do that and that's totally fine, but being versatile with all of that is definitely important.
Yeah, I mean, I second everything you said. The Google Calendar thing is super important. Although I kind of get made fun of, I use Apple Calendar instead of Google Calendar. Most of my friends use Google Calendar, but I think I like the way Apple looks a little better. But kind of scheduling in, I have all of my classes scheduled. I have this meeting scheduled, so I know like this is blocked out in my day. So kind of figuring out, how to block out your day, figuring out you're kind of going to get into a routine, because even though it's not going to be the same every day, you're not going to have the same schedule every day. Your Monday, Wednesday, Fridays might look super similar. Your Tuesday, Thursdays might look super similar. And then week to week, it's going to be kind of getting into that routine for every Monday, it's going to look like this. Every Tuesday, it's going to look like this. I also, at the beginning of the semester, one suggestion I really have is I look at the syllabus for every class. And I have a little document open at all times on my computer that keeps track of every assignment, when it's due, when in the semester it's due, when the exams are, just so I know I'm not missing it. Because in high school, a lot of the time, your teachers will remind you, oh, this assignment's due. Oh, the day before, make sure you turn this in before midnight tonight. In college, a lot of professors don't do that. You're kind of on your own to know when things are getting turned in. to know in advance when the next exam is. Some professors might not even mention the next exam until the class before it happens. So making sure you have that time to prepare and you know that in your schedule, this is when assignments are due, this is when exams are. So that's like one big piece of advice that I would give too as well.
Yeah, I know.
Wonderful advice. Oh, go ahead, Sina.
Sorry, I was gonna say at Capital City, I know they said they have another question. I don't know if you guys want to unmute real quick.
Oh
and-
yeah, we have another question. If there are any, excuse me, are there any organisms that we shouldn't genetically engineer?
Ooh, I think that's a great question. I've done a little research, but I think this might be a great one for our panel that's coming up soon. I do know there are some directions on being able to engineer your different ones that affect the germline, which would, affect everyone down the line from you, where they can't give consent. But I believe our ladies that are going to be in our panel coming up will probably be able to address that question even better than me, on where the ethics of that stand. So great questions. You guys are really rocking it today. You're really making us think hard, and I love it. And I can tell you are really thinking, that next level, applying the learning to what could we use this for? So super jobs. On the screen, you can see just some. Oh, did you have one more question? I'm so sorry.
No, we're good by now. Thank you.
You're good. Oh yeah, and remember to ask them that question when we get there too, if it doesn't already come up in the presentation. On the screen, if you are interested and enjoyed this lab, you guys don't have to wait to college to start getting involved. You can see some of the organizations on the left that you might consider, and then some companies if you want to go ahead and get your foot in the door and do some internships that deal with biomedical engineering, to go ahead and get started on that right away. I'm going to go ahead and share some of the skills that you guys have learned. You learned about bioinformatics, gene therapy. We talked a teeny bit about those medical ethics and what things we should or shouldn't genetically engineer. And you've really gone into the biological databases to help diagnose patients, which I think is such a cool thing, to have that power in our hands to be able to understand things a little better. I am going to shout out to y'all. So you have done such an amazing job, interacting and talking about diseases that you're curious about, brain disease, Parkinson's, sickle cell, Alzheimer's, HIV and AIDS, mad cow disease, and others. And so I want to thank you guys because you're getting more out of it by your interactions. And I love that you're curious. Now that you have these databases, you can start looking for more information about the ones that you said you were curious about. And a big shout out to Mount St. Ursula. I see some girl power going on out there. Some excellent questions for us here in the wrap up. Science Park was doing some amazing things. We've got all these wonderful schools actively learning. So I just wanted to put your pictures up there and show you, we see you, we hear you, and we know you are working hard, and your hard work matters. So I'm going to go ahead and jump us over real quick. I want to thank Dylan and Sinahi for giving us such a great wrap up. Their experience is so helpful. And I want to introduce our amazing powerhouse speakers that are going to be talking with you. You can see Sarah is a graduate of MIT. She studied mechanisms of horizontal gene transfer at the Grossman Lab and developed nanoscale contrast agents for early cancer detection. She currently works as an associate consultant at KX Advisors. And Victoria, she is a senior scientist at CRISPR Therapeutics, specializing in cell and molecular biology. Her research focuses on CRISPR-Cas9, which I think is going to be so amazing. I cannot wait for you guys to hear more about her work. She's authored publications and patents in muscular dystrophy and developing of iPSC-derived immune cell therapies for cancer treatment. Without further ado, I'm going to go ahead and hand it over to these amazing ladies. Thank you guys so much.
Yeah, thank you, Betsy, for the amazing introduction. I was just going to introduce myself really quick, and then I'll hand it off to Victoria to kind of start us off through our presentation. But good morning, everyone. My name is Sarah. I am a recent graduate of MIT, literally just graduated in May, and I majored in biological engineering and business management, and now I work in a more healthcare strategy consulting roles. That means that I'm helping companies like CRISPR, like other pharmaceutical companies, take drugs from the lab to the clinic so that they actually help patients. So I'll get a little bit more into my background later, but for now, I would love to hop it off to Victoria. And Victoria, let me know when you're ready for me to share my screen and we can get started.
Okay, thank you, Sarah. Thank you, Betsy, for an amazing introduction and handing off. Can you hear me well?
Yes,
Okay.
I hear you. You sound
Okay. Sarah, I think you can start with the slides. So indeed, while Sarah is starting the slide deck for me, indeed, I am a scientist at CRISPR Therapeutics and currently specializing in gene editing. Just you can hear from my accent, just like many scientists here, I am a first-generation immigrant here in the US, so that may or may not add some additional complications to any of the research path or any of the professional paths, such as having English as a second language. So I don't know if our students experience any of that, but it can be complicated and fun way of advancing the career too. So Sarah, if you can move to the next slide. There's a lot going on. Before I proceed, because working in industry can be a little bit tricky and what I can and cannot disclose. So full disclosure, everything that I'm going to be talking about now is my personal experience, either educational or research experience, and it's not affiliated with CRISPR, but it's a real life experience that I've had. So as you can see from here, I came from Ukraine. I was born there. I got my full education there, high school, college, grad school, and I got my PhD before moving to the USA for postdoc and then moving to CRISPR, where I am at now. So postdoctoral experience, I'm not sure how much we know about it, but just briefly explain about it. It's a postdoctoral experience that people get to get more experience in different scientific labs after they finish the grad school before they become kind of independent scientists in either academia or industry. It's not required, but it's one of the ways of advancing the career. So moving back to my high school experience, that was quite a journey. Like preparing for this presentation, I kind of went far back and tried to remember how it felt to be a high school student. And I remember myself, not really knowing a lot what to do. Maybe a lot of people are like that. Maybe times changed. But back in the day, it was, I also grew up in a small town, so there was no science fairs or career advisors available to us. So we had to go with the path that was available based on the local colleges or basically what your parents will tell you, what is a good profession for you. So I knew one thing. I didn't want to take my parents' path. My whole family is teachers, and I have great respect for teachers, and I know how demanding this job can be and how much responsibility they carry. But I just knew I would not be able to do that. So this was a big no. And then my mom, of course, wanted me to study economy that I didn't have passion for. And I was good at writing, so I thought I would become a reporter or write for the paper until my dad talked to me and he gave me brilliant advice. He said, why don't you get more deep understanding, more deeper specialty, and then you can, if you still want to be a reporter, you can start writing about that and learn on the job and move on to different areas. And then I consider myself lucky. I met this amazing woman who was a virology professor at the top university in Ukraine. She was a friend of a friend of our family. And when I met her, she told me all about science at the university and how it is to be a college student studying biology. And I just knew that that was it. I always liked the nature, and that just kind of sparkled the curiosity in me. Oh, I'll be able to study nature. How amazing that could be. And so after some additional training, because that was quite a turn for my career decision, I passed the exams and I went to college, and I loved every minute of it. It was hard. I had some catching up to do with other students, but after some hard studying, I became top 5% of my class, and again the life and the science there was great. I was working in parallel, was volunteering in one of the scientific labs at the university, and it was not very well funded lab, so we didn't have any fancy science or very modern science going on there. But if I can take one lesson that I learned from there, was advice from my mentor. He said, never come here in the lab and ask what to do. What do I do today? Ask, why are we working on what we are working? What are the scientific questions? Basically, what do we need to answer? What are we finding here? What experiments we can do to answer those questions? And basically be curious, ask questions every day, talk to your mentors, and that will open the whole world for you. And indeed, so I started reading more literature, started asking questions, and around the time of my graduation, I found another lab that was better funded, and I did kind of joined labs grad school between the university and this better institute for my grad school. And again, I was lucky with my mentors, because not only about the practical science we did, but the conversations we had every day. So we stayed after work and had these debates about every biology and non-biology topic that kind of came to mind, and we had different people coming in and different experiences. So I would say my whole educational experience back in Ukraine, I find more important. The more important for me were the people that I worked with and what I learned from them, the life lessons I learned from them. Mine being in grad school, I went to the US for a couple of months. I volunteered in a couple of labs, went back to Ukraine, graduated, defended my PhD thesis. And by that time, I had 2 job offers for postdoc from Rutgers University. I took one and moved to the USA. I thought I would move for a year or two. It's been 13 years. So I don't have plans of moving back or moving out of the US. So this just shows you how much I liked it here, and it's a great place for science to be innovative, to find every opportunity you can think about. So what did we do? I would just want to use my research project kind of as an example of how we think about science, how we advance science, and the questions we ask, those whys and hows. Sarah, if you can move to the next one. So at Rutgers, we were studying molecular mechanisms of development of muscular dystrophy. And it's a disease that affects mainly boys. And as you can say from its name, muscular dystrophy, it affects muscles, right? So these boys can have troubles walking and then moving. And if you think about muscles, the heart is a muscle, so they have heart problems. The diaphragm is a muscle, so they have breathing problems towards the end of their life. And it's deadly disease. They have short lifespan. And back then and even now, the majority of therapies for this disease are aimed to treat the symptoms, not the cause of the disease. So they help maintain the patient's life and give them better quality of life, but they don't treat the root cause of the disease. So what is it? What is the root cause? Can you go to the next slide? Thank you. And it's can be as small or as big as the mutation in one gene. If you look on the right side, the muscle contains of the muscle fibers, and muscle fibers has muscle cells. And there is one protein here on the bottom. It's kind of labeled as spiral, I guess, banded. It's called dystrophin. When we talk about dystrophin, we think of it as a shock absorber. So that car wheel on the right side, the coiled spring on it, the shock absorber for the car, this is what dystrophin does for the cell. It absorbs the shock, the mechanical shock from the movements. So our cells don't have the bumpy ride every time we move. And when that dystrophin protein is missing in the patients with Duchenne muscular dystrophy, the cells wear out much faster, and the muscle cells die much faster. So there is muscle cell loss and muscle waste and muscle weakness. And why do they not make dystrophin? Why they don't have dystrophin? It's because of the mutations in the patient's DNA. So you know how there is DNA and there are genes. The DNA makes genes, and genes are basically the recipes for proteins, right? So the cell reads the recipe and makes the protein based on that recipe. If there is even one letter mistake, or one letter missing in that gene recipe, the protein cannot be made, or the cell is making dysfunctional protein that cannot fulfill its role. So all we need to do to really get to that root cause of the disease is to fix sometimes just one letter in the gene recipe. It's very easy to say, but how do we do this? And luckily, and that was, at that time, I already moved to UT Southwestern, and around that time, scientific world started talking more about CRISPR editing. Sarah, can you move to the next slide? So it was around 2013, 2014. So CRISPR was known since 1980s or late 1980s, where it was discovered in bacteria. But scientists didn't know what it was for. So they saw it, but they didn't know what the function it was doing until early 2000s, when they realized that this is the bacterial gene editing tool. And only in 2012, two brilliant scientists, Emmanuel Charpentier and Jennifer Doudna, working together in collaboration. One was in Europe, one was in the United States. They worked together to come up with the CRISPR. They developed CRISPR into a tool for scientists to precisely edit genes. Then it was later taken to different types of cells. And I'm trying to count, 7 years later, they got, or 8 years later, they got Nobel Prize for their finding. And it was, scientific world was waiting for this to happen. And another reason why it was important is that it was the first time that that the Nobel Prize was given to a team of two women scientists. I've had a chance to see both of them presenting, and they're truly inspirational women. It's just, I think we are lucky as a world to have them and to have this discovery. And only 10 years later, since it was discovered, we already have the first approved CRISPR-based therapy, which, it's moving with the lightning speed. I know 10 years can be a long time, but it's not long to go from discovery to therapy. So if you can move to the next slide. How does CRISPR work? Maybe you've already known, but I'll just briefly go over it again for maybe a lot of us, a lot of you. CRISPR is a protein molecule that uses a small RNA guide that targets this CRISPR complex to the specific sequence of the DNA that the scientists want to edit. The CRISPR protein makes a cut in the DNA, and the cell then repairs itself, or the scientists can also guide the cell on how, or tell the cells how to repair themselves. Next. So one person, again, that was in 2014, probably, one person in the lab that I was working at UT Southwestern in Dallas, they thought of applying CRISPR to fix Duchenne muscular dystrophy mutations. And to give you a visual how it's happening, on the next slide, we have, I know it's a colorful mess, I'll explain you. So on the left side, again, it's a big colorful mess, but what it is, it's the muscle cells derived from healthy donors, and they're cardiac muscle cells. So green and red, they're just our ways, that's how the scientists are labeled proteins, so we can visualize them. So green is the muscle-specific protein, just to tell us that these are indeed muscles and not liver cells or anything else. So we are working with muscle cells. And the red smears are the dystrophin. You cannot see the molecule, just like I showed you on the cartoon, right? But you can see all together they give us this in our case, the red color. So the normal muscle cells, they have both. They have muscle-specific proteins, they have dystrophin, that's how it should be. In the middle, there are cells derived from the patient with Duchenne muscular dystrophy. You can see green, so they're muscles, but they don't have dystrophin. So we can, even though we stain dystrophin, it's not there, so there's nothing to stain. And on the most right side, it's the same patient cells, but the mutation in these cells is fixed. So these cells, for the first time, start making dystrophin, and that's what we are staining right now. So we see again our beautiful green-red mess in the cells. So that's it's a simple picture, but that meant a world back then. That means that we can fix the mutation and these cells can start making dystrophin that is much needed for the cells to function. And then the truly inspirational point for me, so again, this works, it's all great and it's beautiful, but the truly inspirational point for me was to actually meet patients and their families. We had conferences where families and with the patients came over and they were talking about all of their difficulties in life and how important it is for them to have hope, right? The treatment is still not available to wide range of patients right now, but it's it's developed. So on the next slide, we have from my previous lab at UT Southwestern, we had this very first patient. He's in the wheelchair here. He was the first one to donate his cells for the research, so we could use his cells to see how we can correct the mutation in his cells. So in the left picture, he's with his medical doctor, and the gray-haired gentleman is Dr. Eric Olson, whose lab I was working at the time. And on the right side of the slide, the picture is Eric Olson showing to the patient his own cells, but corrected. So this was, we've talked a lot about this in the lab and how it truly inspired a lot of us scientists working back then. And so this patient, he is a big also DMD patients advocate and truly inspiring person. So that was great. And this was kind of a no return point for me. I knew that I wanted that my research can make a difference. I can make a difference. And I wanted to work in this translational research, gene editing or cell therapies that would make a difference for patients. And with that, with that idea, I moved to CRISPR Therapeutics. And just want to finish up with thank you, with the notion that CRISPR had been applied to so many areas right now, and there's so many diseases that have hope thanks to CRISPR editing tool. And a lot of them are in clinical trials right now. And as I mentioned before, there is one that is already approved for treating the blood disease. So I think this is great achievement for the whole scientific world.
It's incredible.
Yeah.
Okay, awesome. Thank you, Victoria. That was a really, really inspiring story. And I think you guys can really see how, you guys did this in the lab where you kind of see how genes create RNA, create proteins, and how that can affect the symptoms that people are feeling in your daily life. Can you imagine, like Victoria goes in there with her team and she fixes one sort of base pair in the DNA and it has this huge ripple effect on actual people and the way they're feeling, right? I think that's really inspiring. So thank you, Victoria, for going over your journey. I think now I'll kind of introduce myself and I'll go a little bit into my research, which is also on the translational side of things. So by way of brief introduction again, hi, my name is Sarah. I just graduated from MIT and I currently work at a healthcare consultancy. I've been involved with Engineering Tomorrow for a really long time, so some of you may or may not have seen me before. And I was actually the creator of this genetics lab, and I had a great time doing it. I think I did it actually while I was at CRISPR, because I was so inspired by the work that I was doing with Victoria and her team that I was like, okay, everyone has to know about this super cool stuff. So I think we can all thank Victoria for being able to learn about all of this today. So I just wanted to go over a little bit about my journey. It's funny, I've used this same slide for the last 4 years of me being in Engineering Tomorrow, and I feel like people can kind of see it expand over time. But to go over some quick milestones, when I was in high school, I started doing really basic research, just kind of focusing on problems that were really close to home. So the way I got into research, my mother is a big gardener. She's really, really into plants, and we always kind of grew her up around those plants. And I remember one spring, she had spent all this work trying to grow a set of daffodils in our garden, and none of them actually ended up blooming that spring. And I remember she was devastated. So what I did when I was a kid was I was like, hey, there must be something wrong in the garden. Let's see what's wrong. So I went to the store and we got 3 different kinds of fertilizer and we tested 3 different kinds of fertilizer in different parts of our garden. And we found out that the fertilizer she was using before actually did not work in our type of soil. So when she switched to the kind of fertilizer that we identified in our experiment, her plants grew like crazy. So that's when I was like, at a very young age, I realized that this process of inquiry, this process of testing and testing different things can actually have an impact on your life right now in a very real way. So I started more with these local problems, like helping my mom in the garden, helping the algal blooms that were in my grandmother's pond, trying to figure out biological solutions for those problems. And I fell in love with this idea that you can use biology, leverage biological mechanisms to combat some of the world's biggest challenges, everything from climate change to antibiotic resistance to cancer, to Duchenne muscular dystrophy, everything like that. So when I got to MIT for my undergrad or college, I was really interested in investigating fundamental biology. So this is the idea of like looking at the underlying mechanisms of how things work. And what I was doing was I was looking at how bacteria exchange genetic material to give themselves traits that they didn't have before, right? So if you guys have ever heard of the concept of antibiotic resistance, this means that once bacteria have been exposed to an antibiotic, they kind of genetically develop a way to survive that antibiotic And what's really interesting is that they can give that trait. They can give that trait of resistance to their siblings. So this is like if I were to shake Betsy's hand and I gave her the trait for having black hair, right? And then just then and there she developed black hair, right? So we can't do that. That would be really cool if we could, but bacteria can do that. So I was working in that lab for a couple of years. And it was when I was working in that lab that my mother actually was diagnosed with endometrial cancer. And this was when the idea of, having to develop a biological solution to a problem urgently really came to the forefront for me. And it was when I realized that this world really needs more smart people to tackle these types of diseases that really affect everyone. Like, I think all of us have a family member that we know that has been touched by cancer, that has been touched by another chronic illness. And this was when it was like a wake up call for me. It was like, I can't just do the fundamental biology research. I have to do this more translational side of things. So that's when I moved to CRISPR. I joined Victoria's team in the regenerative medicine department. We were working on a lot of cool stuff. There was like my first taste in translational medicine. Victoria remembers, like, before I came into that lab, I had only worked with bacteria, and like the idea of working with a human cell after that was like, it was very different. But that summer was like a crash course in how to deal with human cells, and I learned a lot, and I really liked it. So afterwards, I came out of that knowing that I kind of want to do more translational research and type of research that Victoria and her team were doing. And I also wanted to find ways to contextualize how that research actually gets into the hands of patients like my mother, right? So that's when I joined another firm. This was like a healthcare company creation firm called Aegis Ventures. We were creating healthcare companies from scratch. So if you guys ever want to look it up, I worked on a company called Upliv, UPLIV. It was a company developing digital health solutions for women going through menopause. And we also developed some solutions for women's oncology, so types of cancer that my mother was grappling with. I learned a lot there, and I kind of learned this idea that the solutions we develop in the lab need to be really impactful and actually help people. So that's when I moved into this lab, the Laboratory for Multiscale Regenerative Technologies at MIT's Koch Institute for Integrative Cancer Research. This is the research that I'm going to talk a little bit about today. But I'm also happy to talk about anything else. I did my senior thesis in more women's health related stuff, and I'm very passionate about that as well. So if anyone's interested in that, feel free to ask Betsy for my contact information. We can get offline and talk about it. But I just wanted to show some quick pictures of my journey so far. So this is the kind of research I was doing when I was in high school. So this is like when I was in the same place as you guys. I didn't do research in a big fancy lab at a university. I did research in my basement using bins that I bought from Target on discount. And this is all duct tape. And these are lights that we found at the junkyard that we hooked up to my ceiling using my mother's garden twine. So this is the idea that you can kind of investigate problems regardless of the kind of resources that are available to you at the time, right? Like Victoria was saying, in her lab back in Ukraine, like they didn't have the most resources, but she was, you make do with what you have and you ask the questions that you want to ask. You figure out ways to answer those questions. So that's one little tidbit I'll give you guys, like, regardless of what kind of state you're in, don't ever think that because I am 16, because I'm 15, because I go to a high school that is not a STEM high school or whatever, that you can't do research, because you for sure can. When I got here, I moved into the biology department, went to CRISPR. I also have some pretty sustained cells to look at, although they're not as pretty as Victoria's pictures. And now I work at KX. So I'm going to talk a little bit more about this research, and this research was particularly on early cancer detection. So the idea is improving liquid biopsy for early cancer detection. And I'll go into more details about what liquid biopsy is in a second. But first, I want to start off with, why is cancers like my mother's cancer, why are they so hard to treat? So I'm going to go over kind of three reasons. So the first reason is that it's heterogeneous. It's different when it goes from patient to patient. So you think of cancer like a weed in a garden, right? just like weeds, cancer comes in many different types, which makes it heterogeneous. It makes it varied. And these different types of cancer can respond to treatments in different ways, just as certain weeds might respond differently to different kinds of weed killers. Now, imagine if the weeds could spread seeds to different areas of the garden. And as they always do, people who have a garden, you know that, like they always do. So cancer can do something similar. It can spread, it can metastasize to other parts of the body. And this spreading makes it much harder to get rid of because it's no longer just in one spot. You have to treat the cancer in all these different kinds of spots. So continuing with our little garden analogy, imagine that some of the weeds in your garden that have spread to all these different parts of the garden start to become resistant to the weed killer that you're using. Over time, these weeds will adapt and they'll no longer respond to the same chemicals that used to kill them really easily. So this is how cancer cells can also become resistant to chemotherapy, can also become resistant to the ways in which we treat them. So as these weeds evolve, as this cancer evolves, we need to develop different, stronger treatments to control them. And they may require completely different mechanisms to actually remove them from the body. So doctors need to constantly find new drugs and new treatments to effectively target these adapted cells. So the ability of cancer cells to learn and almost outsmart the treatments adds to the challenge of curing cancer, especially after it's spread. But when cancer is only in one place, right, like a weed confined to only one part of the garden, it's way easier for us to treat and remove. But if it has spread to too many areas and it's been allowed to spread for too long, finding and effectively treating all the spots becomes a lot more difficult. So this is why cancer is so tough to deal with when it's in its late stages, when it's kind of spread to more distant areas. And if we can detect it early, the survival rates are a lot better, right? So you can see here on the graph on the right, this is melanoma, type of skin cancer. And if you detect it in stage 1, the curing rates, like the rates in which we can cure that cancer, are actually extremely high, right? And that's a kind of a similar trend that you can see across all cancers. The idea is that if you can detect cancer early, you have a better chance of treating it. So now the question becomes, how do we detect cancer early? Well, there's two kinds of ways in which we can detect cancer. First, there's biopsy, and there's also CT scans. So a biopsy involves taking a small sample of tissue from the body and looking at it under a microscope to see if there are cancer cells. And a CT scan uses specialized X-ray equipment to create detailed images of inside the body, which helps the doctors kind of visualize where the cancer might be. But both methods have difficulties in catching cancer early. So biopsies, for one, they're very invasive. You have to actually open someone up to get that sample, which is not always possible, and it's not always safe, especially if the cancer is in a part of the body that's really hard to reach. And then the other thing is, because biopsies only take cells from a small area, you can imagine that there's a chance that they might miss the cancer cells if those cells just aren't in the sample that they take. Now, CT scans, they're definitely less invasive. They don't require taking tissue, although they use a lot of radiation. They might not show really small early stage cancers clearly, just because they're so small, they're not visually detectable on the scan that people might look at. They're kind of better at finding larger, more developed cancers, but they might not pick up on the tiny early ones. So because of these limitations, both biopsies and CT scans can oftentimes miss early stage cancers, which really limits our ability to treat cancer at the early stages. And people are kind of thinking, like, how can we do this? And you can imagine, when you go to the doctor for your annual physical and they want to screen you for a bunch of diseases, what is the first thing they do? They send you to a LabCorp, right? They send you to a lab to get a blood test, right? So what about a blood test? Why can't we do a blood test for cancer detection? So this is the idea behind my research. And in order to do a blood test for cancer, you need something that is called a biomarker. So a biomarker is a special marker or a sign that doctors can use to learn more about a patient's health. So think of it kind of like a clue, like similar to the clues you guys looked at in your lab today, the clues of symptoms, the clues of what genes the people have. Think of it as a clue that helps a doctor figure out if someone might be sick, how severe their illness is, or how well their treatment is working. And biomarkers can be found in the blood, they can be found in the urine, or they can be found in any other kinds of body fluids. And they can be things like proteins, they can be things like genes, or even other molecules. So for example, if someone's blood test shows high sugar levels, that indicates to us that they might have diabetes, right? So you can kind of do this, measure a biomarker, and then make some sort of conclusion based on that biomarker. So we need to find a biomarker that can tell us that can indicate to us that there is an early stage cancer. And the biomarker are these things. They're called circulating tumor DNA, or ctDNA. So every tumor, when they're growing and developing, honestly, every cell in the body, they are constantly shedding pieces of DNA called cell-free DNA into the bloodstream. And tumors produce a special kind of cell-free DNA into the bloodstream called circulating tumor DNA. And these are the things that we want to measure. So the idea is that if you can measure the amount of circulating tumor DNA in the bloodstream, you can see if there is a level of early stage cancer. So we could even get to a point where you're going for your annual physical and your doctor says to you, I'm going to send you for a blood test and we're just going to test you to see if you have any cancer. And we can get that really early stage, regardless of whether you have symptoms or not. But the problem here is that when people are taking a blood test, you're going to get a mix of circulating tumor DNA, which is the DNA you want to measure, and also cell-free DNA, right? Like just normal DNA that's being shed by cells in your body. So how do you figure out the difference between these two cells, right? When you're measuring this DNA, how do you figure out which one's a circulating tumor DNA and which one's a cell-free DNA? Well, I think you guys know the answer to that after going through your lab. So you want to figure out what is the difference between this one and this one. You use DNA sequencing, right? And you see where in the DNA sequence are the differences in these two strands. And you can figure out when you sequence, there's actually this difference, right? Just a difference of 3 base pairs can make a massive difference in whether this is a circulating tumor DNA or a cell-free DNA. So in the case of our circulating tumor DNA, the sequence that we're looking for, the sequence that's different, is called a wisdom sequence. So what we do is we take a blood draw, we measure this circulating tumor DNA, we put it through a DNA sequencer, and we kind of look at whether it has this different sequence than a cell-free DNA sequence, whether it has this wisdom sequence. And if it does, to a certain degree, we can conclude that there is some sort of early stage cancer. And we can even tell you what kind of cancer it is and where it is. Super cool, right?
It's amazing.
Yeah, super cool. But there's still a problem. So there are these cells called macrophages in our body. And imagine they're like the cleanup crew of our body, right? They're the security guards that are patrolling our bloodstream to make sure that there is nothing going on. So they have 3 main roles. They're garbage collectors, right? They find and they eat dead cells, bacteria, other debris that can exist in your body. They engulf bacteria and stop infections before they spread. And beyond just cleaning up, they also are alarm raisers. They can alert the immune system to infections, to send out signals alerting other immune cells to rush to the site of infection and help clean it up really quickly. So this helps us mount a faster and more effective defense against diseases. And the last thing they do is they are involved in repairing the body. So after an infection or an injury, a scrape, they can help to clean up the area by swallowing dead cells and debris. And they also release substances that encourage new tissues to grow and aid in the healing process and restore normal function. So this is kind of a diagram here, but you can imagine when macrophages are going around the body and they're eating all this stuff, they're also eating the cell-free DNA in the body, which makes it so that we can't actually measure it anymore. So they surround these pieces and they gobble them up to keep our body clean and healthy, but which is good for the sake of our body, but bad for the sake of our technology. So what we did was we developed a nanoparticle that would be uptaken by these macrophages to slow the uptake of cell-free DNA for a certain amount of time. So the idea is that before you go in for your blood test, we're going to give you this nanoparticle treatment. We'll inject it, we'll wait an hour, and then we'll take a blood draw, and we'll actually be able to see the mutations that are detected. And this is a cute picture that I found online of our research. So you can imagine this is like our nanoparticle here with the shield. This is our ctDNA that we're trying to protect, and the nanoparticle essentially prevents the macrophage from eating our cell-free DNA. And we found out that this works really remarkably. So you look at this graph on the right here, the more nanoparticles that you're injecting into the bloodstream, the more cell-free DNA, the more circulating tumor DNA you can detect, right? It's going up. So this technology is so successful that it's actually being turned into a company called Amplifier Bio. And I put a QR code here for you guys to scan and read more about it if you're interested. It's super cool, very, very new technology. It literally was just published this year. And I think this is going to make a really big difference in the way in which we diagnose cancer. But that's just an example of how the stuff you guys are learning today is actually being translated, both in terms of Victoria's work with gene editing and also in terms of my work with diagnosing early stage disease. So
Wow.
I'm not sure how much time we have left, Betsy, but
I have a couple questions for you. I have a question from Whittier Christian High School as well as Capital High School. And they were asking about genetic engineering. Is there a way that genetic engineering can prevent inherited diseases? And also, are there certain organisms that we should not genetically engineer? You two ladies can answer those amazing questions.
I can start, Sarah. Feel free to to add. They're actually very, very related, both of these questions, because there are things that we can do and there are things that we should not do, probably, right? So genetic diseases are and it's a really bad situation, and at this point we can screen for them, right? We can know that whether the kids may or may not have it. But I think, Betsy, as I heard a little bit of of your answer, and this was right on spot. Like that there is a huge ethical concern about editing the germlines and future human babies, because there is even though we study CRISPR a lot, it's still a new technology, and we don't know how it's how it works exactly, and maybe we don't want to know on the editing germlines lines level. So I think scientists are working hard on treating or fixing the genetic diseases in adult patients, but safety comes first. So kind of from the experience, I know that the first questions that the FDA is asking, is it safe? So there is, it's all fun to see how we can fix the disease, but there is a huge concern about this, what we call off-target targeting by CRISPR. So scientists are doing a huge amount of work to make sure that there is no accidental cuts somewhere else in the genome too. So in addition to concern about editing that specific mutation, inherited mutation, there's a big concern about creating other mutations somewhere else in the genome. So scientists are using whole genome sequencing and thoroughly looking through the whole genome of the edited cells to make sure that there is no off-target editing and accidental breaking other genes that could be transferable to the kids, to the next generation. I hope that's it.
That was such an incredible answer. And it looks like Renaissance asked a couple of follow-on questions to that. So I'll ask that in kind of a general term, and then we'll wrap up. So Victoria and Sarah, they were asking, are there any other side effects that happen when you do the CRISPR editing? And also you had talked about the metastasis of cancer. Like, how is that happening? Is it spread through food? How does it get to the other parts of the body? I know those are a little unrelated, but I wanted to throw those in there before we wrap up.
Yeah,
Yeah, maybe Victoria, you can start, and then I'll take the metastasis.
Okay. Okay. Yeah, so I mentioned in my previous answer, one of the side effects and undesired side effects of CRISPR is can be that, you know, we are using a very small, short spaced guide RNA that can have, and the genome is so huge that that there is a chance that that same repeats, that the CRISPR system can recognize some other part of the DNA as the site that it needs to cut. So that's what we call off-target editing. So it's the editing in the non-desired site, not the targeted site. But again, being in industry and academia, we are screening hundreds of different this guide RNAs to make sure that we are selecting for the one that is clean and is fitting only to one part of the genome, and there is no accidental cuts. So that's a safety, and that's a main concern.
Wow.
Yep. And then quickly for the other one, I know we're up on time, but I think the way that I guess generally it works, and you can also do some more research online, but these cancer cells will break away from the primary tumor, and the primary tumor kind of establishes itself through a lot of different reasons. So sometimes there's a mutation that occurs that allows a cell to grow more than it's supposed to grow. Usually cells have signals that tell them to stop growing or tell them to kind of initiate cell death when things are going wrong, but cancer cells often don't have those mechanisms in place to tell them when to stop. So when a cancer starts developing in one area, there can be other mutations that start telling the cancer cells in that area to stop adhering to that one spot. And then a cancer cell can enter either a nearby blood vessel or a lymphatic vessel. And once it enters those systems, it can spread throughout the body through the bloodstream, through the lymphatic system. Some of them face challenges while they're in the system. They're undergoing things like shear stress, but to survive, they might form clusters, and that's how they end up in different parts of the body. They can attach to different parts there, and I think that's there's definitely more intricacies involved, but that's the short answer.
Wow, you ladies, I feel so honored and blessed to have gotten to listen to you both. Some key takeaways that I heard from you is never ask what I should do today, but ask why are we working on this and how can it be beneficial? Such good advice. And that testing and analyzing problems around us right now can change our life and our surroundings today, that we don't have to wait to start analyzing these things. And realizing that our work makes a difference. And so thank you, ladies, so much for being here with us today. It has been such a pleasure getting to hear your experiences and that you're willing to just to take time to share them with us. So thank you so much. I'm going to hand it back over to Monica for the final steps here.
Well, I want to say thank you to Betsy for leading such an amazing day, but thank you to all the teachers and students who made it possible here live today to